Hello, my name is Candace Ward. This video will delve into the mechanisms of transmission of neural impulses. We'll figure out how neurons talk to each other and how those signals travel from one end of the neuron to the other. We'll start off by defining and exploring the chemical basis of the action potential and how these signals propagate down an axon. Then we'll take a look at what goes on in the synapse and finish up with a brief look at how neurotransmitters convey information from one neuron to the other. In this video, we'll aim to establish a firm understanding of how neurons transmit signals via action potentials and neurotransmitters. By the end of this video, you'll be able to answer questions like the one at the bottom of the screen that ask about specific phases of an action potential. Neurons transmit information from their cell body down to the presynaptic terminal through an electrical signal called an action potential. These action potentials are called all or nothing. Either you get a signal or you don't. There's no such thing as a small action potential versus a large one. It's a binary system. Let's follow the trail of a signal in our neuron. As you probably already know, a neuron receives information in processes called dendrites, here on the left side of our figure. That information travels to the very edge of the axon, an area known as the axon hillock. Here's where our action potential actually begins. The action potential starts at the axon hillock, then travels down the axon to the presynaptic terminal. However, the MCAT is going to ask us more detail than just the path of an action potential. The test makers want you to know exactly how that electrical impulse travels down a cell. To understand that, we need to figure out what membrane potentials are. A resting membrane potential is a measurement of the electric potential difference across a membrane. Specific concentrations of ions help establish this potential difference. A transmembrane protein pump, sodium-potassium ATPase, pumps sodium and potassium in opposite directions across the neuron membrane in order to create this potential difference. Let's take a look at how ions establish this difference. Up top will be the outside of our neuron, and below the membrane will be the inside of the cell. Outside the cell, we have lots and lots of sodium ions, indicated by the little green triangles, and very few potassium ions, which are the orange squares. When our sodium-potassium ATPase is working, it pumps 3 sodium ions out of the neuron for every 2 potassium ions that are pulled into the neuron. If we pull 3 positive ions out of the cell and 2 positive ions of the same charge into the cell, we'll end up with more positive charges outside the cell. Inside the cell, we have the opposite concentrations. There are more potassium ions inside the cell than outside, and there are fewer sodium ions in the cell than outside it. Due to that ATPase, we know that we pulled more positive charges out of the cell than were put in, so the inside of the neuron has a net negative charge. The difference in charges outside the cell versus inside it establish the potential difference. The commonly accepted value for the resting potential of a neuron is negative 70 millivolts. I'm specifying a neuron at rest here because this membrane potential changes dramatically during an action potential, as we'll examine next. As we just established, the resting membrane potential is negative 70 millivolts. Without any stimulus, the neuron will hang around that membrane potential. However, once we start getting some information in from the dendrites, the membrane potential will rise. Once it gets to about negative 50 millivolts, we get an action potential. Negative 50 millivolts is the commonly acknowledged value for the action potential threshold. This means that the membrane needs to reach this potential in order to get an action potential, since action potentials are all or nothing. Now let's see how the membrane potential changes during an action potential. Once we get some stimulus, the membrane begins to have a more positive charge. Voltage-gated sodium channels open up when the membrane becomes more positive. And you probably already know that molecules like to flow down their concentration gradients. Earlier, we mentioned that there's a huge sodium concentration outside the cell versus inside. So, where do you think those sodium ions will flow once those sodium channels are open? Into our neuron. So now we have a flood of positively charged sodium ions pouring into our neuron, causing our membrane potential to shoot all the way up to about positive 35 millivolts. When the membrane potential increases, moving away from the resting membrane potential, we call that depolarization. This depolarization is caused by sodium ions entering the cell. And now our neuron has a membrane potential around 35 millivolts, which signals our slower moving voltage-gated potassium channels to open. At this point, the voltage-gated sodium channels close, so our membrane potential won't increase anymore. But now we have those potassium channels open, and there's more potassium inside the cell than out, so those potassium ions rush out of the cell. As you'd expect, losing positively charged ions makes our membrane potential drop again, heading back towards that resting potential. When the membrane potential becomes more negative, approaching the resting membrane potential, we call this repolarization. This repolarization is caused by potassium ions flowing out of the cell. But our potassium channels are both slow to open and slow to close, so we overshoot the resting potential a bit. This overshoot is due to potassium ions flowing out of those still open potassium channels. When the membrane potential becomes more negative than the resting potential, we call this hyperpolarization. And finally, as we can see on our graph, the sodium-potassium pump reestablishes the correct sodium and potassium concentrations inside and outside of the cell. So our membrane is happily sitting at negative 70 millivolts again. Now we have a better understanding of exactly what's going on in an action potential. It's a change in membrane potential due to ions flowing down their concentration gradients. But that's only happening at one specific spot on the membrane. This signal has to travel all the way from the axon hillock to the presynaptic terminal. Even though we'll cover the propagation of action potentials in intimate detail, remember that these action potentials are occurring extremely quickly. So, once our action potential travels from the axon hillock to the presynaptic terminal, neurotransmitters are triggered to be released into the synapse. So that happens way at the end of the neuron, but how does it travel from one end to the other? Let's take a look at this diagram and see if we can figure it out. Here we see an action potential occurring on the left side of this membrane, colored green. We noted earlier that a positive membrane potential causes sodium channels to open so that these sodium ions could flow in, depolarizing the membrane. If this membrane is now positive, what do you think the sodium channels in the adjacent portion of the membrane are doing? They're starting to open too. So the depolarization of one portion of the membrane causes the next portion of the membrane to depolarize, sending the action potential down the axon to the nerve terminal. But if the portion of the neuron on the right is becoming more positive, wouldn't the membrane to the left depolarize again? Nope, something called a refractory period occurs after repolarization, preventing the neuron from depolarizing. There are two types of refractory periods, the absolute refractory period and the relative refractory period. The absolute refractory period occurs when the sodium channels inactivate and refuse to open. If these channels refuse to open, no depolarization can occur. The relative refractory period occurs when the cell is hyperpolarized and potassium channels are still open. During the relative refractory period, the membrane can still depolarize, but it has a much higher threshold, since the cell has a membrane potential lower than the resting potential. And the last type of voltage-gated channel that opens during an action potential is the voltage-gated calcium channel. These channels are typically at the far end of the neuron by the nerve terminal. The opening of these calcium channels triggers the neurotransmitters to be released into the synapse. As we're talking about the propagation of these action potentials, it's important to note that in myelinated neurons, ion exchange occurs at the nodes of Ranvier, not in the myelinated portions of the axon. We've covered all the processes that occur during action potential propagation, but let's go a bit more in depth about those thresholds and what controls whether we get an action potential or not. Neurons can receive both excitatory and inhibitory input. Depolarization of a neuron initially occurs due to excitatory input. Our membrane is becoming more positive, approaching the threshold for the action potential. With excitatory input, we have a greater chance of getting an action potential. Hyperpolarization of a neuron can occur due to inhibitory input. Since our cell has such a negative membrane potential, it's going to take a lot of stimulus to get our membrane up to that threshold value, so action potentials are far less likely. Lastly, we have summation. Summation occurs when we have multiple stimuli coming into a single cell. These stimuli add together to hopefully get our receiving neuron to an action potential. There are two types of summation, spatial summation and temporal summation. Spatial summation occurs when multiple neurons are synapsing onto one receiving neuron. These other neurons are each stimulating the same neuron in an attempt to either excite or inhibit the postsynaptic membrane. Temporal summation occurs when one neuron sends multiple rapid signals to the same receiving neuron. These signals add together to get the receiving neuron to the threshold value for an action potential. This is a good time to mention an important fact for test day. Multiple stimuli, or a particularly strong stimulus, does not result in an extra strong action potential. Instead, a stronger stimulus can result in an increase in the number of action potentials in the receiving neuron. We've worked hard to get to this point, the synapse. We traveled with our action potential down the axon, and now we're at the place where an electrical signal becomes a chemical signal. The neuron takes the electrical impulse of an action potential and translates it into a chemical signal with neurotransmitters. This change happens at the synapse, the space in between two neurons. Up top, we have our presynaptic neuron, which is the neuron sending the signal, and below we have the postsynaptic neuron, which receives the signal. Way at the top, we have our action potential, which has traveled quite a long way to get here. When this action potential reaches the presynaptic terminal, it triggers the release of neurotransmitters into the synapse. Prior to release, neurotransmitters are stored in membrane-bound vesicles in the presynaptic terminal. When the action potential reaches the nerve terminal, voltage-gated calcium channels open and calcium flows into the cell. This influx of calcium signals the vesicles to fuse with the presynaptic membrane, releasing neurotransmitters into the synapse via exocytosis. The neurotransmitters will then travel across the synapse and bind to receptors on the postsynaptic membrane, where they will have an effect on the second neuron. Our neuron has successfully transmitted its signal, but what happens to those neurotransmitters floating around in the synapse? If these molecules remain in the synaptic cleft, they'll continue to trigger the postsynaptic membrane, unintentionally creating another signal. So we need to get rid of these molecules. One method is to chop up the neurotransmitters with enzymes. Acetylcholine is a neurotransmitter, and once it has done its job, acetylcholinesterase jumps into the synapse and chews up the molecules to prevent further stimulation of the receiving neuron. A second method to get rid of neurotransmitters in the synapse is through reuptake. Reuptake carriers work to pull neurotransmitters back into the presynaptic terminal. Reuptake actually performs two jobs. In addition to removing neurotransmitters from the synapse to prevent continued stimulation, reuptake allows the neurons to conserve energy and resources, since the neuron can reuse these same neurotransmitters during a later signal. As a medical student, you'll learn all about reuptake carriers. You've probably already heard of a common one, SSRIs. SSRI stands for Selective Serotonin Reuptake Inhibitor. In patients with mental health disorders, SSRIs can help prevent serotonin from being reabsorbed back into the presynaptic terminal. This allows the neurotransmitter to continue stimulating the postsynaptic membrane and compensate for the patient's depression. Other neurotransmitters that are commonly reuptaken are dopamine and norepinephrine. Another common regulation method is simple diffusion, where the molecules just drift away from the synapse. This happens very frequently, but the other two methods, destruction by enzymes and reuptake, are tested far more often on the MCAT. We're done with content for this video. Let's test our knowledge with a couple of practice questions. This one asks us about hyperpolarization. Take a second and pick an answer. The answer is B. Hyperpolarization occurs when the potassium channels are still open, so the membrane potential is lower than the resting potential. At this point, we get a refractory period, since it takes a lot more stimulus to get the membrane potential up to the threshold in order to achieve an action potential. Now we're asked to explain why neurotransmitters are removed from the synaptic cleft. Take a second and pick an answer. The correct answer is C. We don't want those neurotransmitters hanging around after they've already transmitted their signal. This allows for discontinuous signaling, which means that there are breaks in between signals. We explored some really cool aspects of neural transmission in this video. Information travels down an axon through an action potential, which is the exchange of ions across a neuronal membrane. This action potential travels all the way to the presynaptic membrane, where it triggers the release of neurotransmitters into the synapse. And once those neurotransmitters travel across the synaptic cleft, they bind to receptors on the postsynaptic membrane and cause the receiving neuron to either continue the signal or inhibit firing. The transmission of neural impulses is super interesting, but it can sometimes be confusing. So feel free to come back to this video as often as you need in order to solidify these concepts for test day. Thanks for watching.